Hello, my name is Nish Kumar, and I'm a Senior Global Product Manager here at Bio-Rad. Today, I will be talking about the SEQuoia Express Stranded RNA-Seq kit. It's a very novel 3-hour, 3-tube workflow for comprehensive transcriptome profiling. A typical next-generation sequencing workflow happens in multiple steps. The first starts with experimental design. So here's where the replicates, the number of samples, keeping in mind the data output, the platform that is being used is being discussed and decided. Once the experimental design is fixed, based on that, the samples are chosen, and usually, depending on the sample type, requires some amount of sample preparation. For example, if it's FFPE samples or cells, extraction of the total RNA from those sample types usually takes about half a business day to 1 business day. And the time taken kind of varies with sample. So once the samples are prepped, usually it results in a total RNA output. Next comes a very crucial step of library preparation. The library preparation is where the total RNA is converted to sequenceable library molecules. And this is very important because this step retains the complexity of the biological material, but also making it easy to sequence. So today we will be discussing and looking at the SEQuoia Express workflow and how it can help alleviate some of the pain points created in a typical RNA-Seq library prep workflow. A typical workflow takes about 6 to 10 hours and can usually be pretty time-consuming and hands-on. Once the libraries are obtained, the libraries are sequenced on a sequencer. And depending on the throughput of the sequencer, it typically takes anywhere from 12 to 24 hours. So it could be an overnight run to a bit longer. Once the data is obtained, the FASTQ files typically have to go through a data analysis workflow. So depending on the bioinformatics expertise of the person doing the analysis, the workflow can be pretty time and labor intensive. So we covered the next generation sequencing workflow from sample to obtaining final results. The library preparation and data analysis are very crucial and critical steps that play a very important role in getting biologically significant data from your samples. So we will look at these two in detail and see how Bio-Rad solutions help alleviate the pain points in the NGS workflow. So introducing an RNA library preparation method using SeqZyme. SEQuoia Express RNA Library Prep Kit is a 3-hour, 3-tube library construction workflow. So typically we start with total RNA. The total RNA undergoes fragmentation. We use a proprietary enzyme called as SeqZyme, and we'll talk more about the benefits of SeqZyme on the next slide. So SeqZyme is an enzyme that does a continuous synthesis that is better than ligase. So it's a very high fidelity enzyme that is better than ligase. SeqZyme does the RT reaction and attaches the adapter on the 5' end, producing a cDNA. The cDNA then undergoes PCR amplification, where the sample-specific indexes are attached. After this, you can proceed to ribodepletion and use the SEQuoia ribodepletion kit to remove unwanted rRNA fragments. So as you can see here, this novel workflow, because of SeqZyme, happens in 3 simple steps. So the workflow time from here to here takes only 3 hours. So let's look at SeqZyme. SeqZyme is a proprietary engineered retrotransposon. So SeqZyme enables the single continuous synthesis reaction that we saw in the workflow previously. SeqZyme does not require any hybridization or template-based addition, which makes the protocol time very fast. SeqZyme is highly efficient, better than ligase, so due to the high conversion efficiency, fewer PCR cycles are required. Hence, there is reduced amplification bias. SeqZyme is also very robust. It can accept low to high inputs. The SEQuoia Express workflow goes from 1 nanogram up to 1 microgram. And we'll look at some data on the upcoming slides, but SeqZyme does not have any bias. So it enables unbiased capture of the transcripts present in your sample, thereby delivering more information. When samples with different input ranges are run through the SEQuoia Express pipeline, we see very high concordance between the replicates as well as the input range. So let's look at some data of the 10,000 most highly expressed transcripts across the different input ranges. So on the first graph here on the left, between 1 microgram and 100 nanogram, we see the concordance to be 0.97. And this trend continues irrespective of the input ranges that we compare against. The same holds true for 10 nanogram versus 100 nanogram, or 1 nanogram versus 100 nanogram input. So irrespective of the input range that you go in with for the SEQuoia Express, you get the same data out of it. So what does this mean? This means that for samples that are available only in limiting quantities, you can still use that to process your RNA-Seq library using SEQuoia Express and compare it to data that was obtained using a higher input range. So irrespective of the input range, you will get same information and you can compare the results between the two. SEQuoia Express produces robust, reproducible, and unbiased data. So here on the left, we see the SEQuoia Express number of genes detected being compared to Kappa, NEB, and Illumina. We see that the number of long non-coding RNA and mRNA genes detected is comparable across the different workflows. So SEQuoia Express can be used and any of these workflows can be replaced and you will get a similar number of genes detected. On the right here, the 5' to 3' transcript coverage across the input range from 1 nanogram to 1 microgram is very even. This indicates that irrespective of the input range, you will get a very even, complete transcript coverage across your transcript of interest. The data that we looked at so far was run through an optimized data analysis pipeline that has been specifically designed to fit the SEQuoia Express workflow. The data analysis pipeline accepts FASTQ files, does the pre-processing, alignment, deduplication, and counts the long RNA and mRNA transcripts, does the normalization, and reports out the data in easily readable formats. This easy-to-use data analysis pipeline comes in two versions. So the first one is the SeqSense Analysis Toolkit, which is a downloadable version for integration with local infrastructure. The SeqSense web app is an online application where you can upload your FASTQ files and the data is processed on the cloud and you get the same reports as you get from the analysis toolkit. So depending on the expertise of the user, you can pick either one of these ways to process your data and still end up with the same results and same set of reports. So to summarize, we looked at the SEQuoia Express RNA-Seq library prep kit. So what are the benefits of this kit for transcriptome analysis? This kit is a short 3-hour 3-tube workflow. It is very automation friendly, very easy to use. It comes with unique molecular tags for identification of PCR duplicates. It is also provided with an optimized pipeline for secondary analysis in the form of SeqSense. So if you're looking to detect changes in cancer transcriptomics or to do biomarker and drug target discovery, or you want to uncover gene targets for genetic disease research and more, SEQuoia Express is the solution for you.